So now let's talk about the Calvin cycle, the second step of photosynthesis. So recall from yesterday's lesson that the energy captured in the light-dependent reactions as ATP and NADPH powers the production of carbohydrates from CO2 in the Calvin cycle. And this occurs in the stroma of the chloroplast, so that outer space. Carbon dioxide will diffuse through the stomata into the stroma of the chloroplast. Now, recall from the leaf anatomy lesson that stomata are the openings in the lower epidermis of the leaf and are controlled by guard cells. So those guard cells can open and close, allowing CO2 in, oxygen out. Then there's that kind of spongy mesophyll area where it's the cells are a little more spread out, allowing for those gases to diffuse from where they are to where they need to go. The reactions of the Calvin cycle are kind of organized into three basic stages: carbon fixation, reduction, and the regeneration of something called RUBP, which you kind of got an idea of what that is from the POGL. And I'll explain more about RUBP as we proceed through this. So in addition to carbon dioxide, the stroma contains an enzyme called rubisco. Okay, that's the abbreviation. It's got a big long name that nobody cares about. We all just call it rubisco. So it contains an enzyme rubisco and this other molecule called RUBP, which has 5 atoms of carbon as well as oxygen, hydrogen, and a phosphate group on each end. So step 1, which is carbon fixation, rubisco will catalyze or speed up a reaction between carbon dioxide and this RUBP molecule, which will then form a 6-carbon compound that is immediately converted into 2 3-carbon compounds. This is called carbon fixation because the carbon dioxide is fixed from its inorganic form into inorganic molecules. So yes, carbon dioxide is technically an inorganic molecule, and it's going to be fixed into organic molecules, which we know are glucose. In the second step, reduction, ATP and NADPH use their stored energy to convert the 3-carbon into another 3-carbon compound. This is a reduction reaction because it involved the gaining of electrons by molecules. And then the molecules of ADP and NADP resulting from the reduction reaction, right? They've donated their electrons, return to the light-dependent reactions to be re-energized, which means they're going to return and bring more electrons to the Calvin cycle. Then in the third basic stage, the regeneration of RUBP, 1 carbon atom from the converted 3-carbon molecules will leave the Calvin cycle. Man, that bell's long. To contribute to the formation of glucose or C6H12O6. Because glucose has 6 atoms, it takes 6 turns of the Calvin cycle to make 1 glucose molecule. Then the remaining 3-carbon molecules regenerate RUBP, which then enables the system to prepare for the carbon fixation step. So the cycle repeats. And ATP is also used to regenerate RUBP. Now, the names of these RUBP and these 3-carbon compounds, those official names are above and beyond the scope of the course. We just need to understand the general things that are happening in the Calvin cycle. So rubisco will catalyze a reaction between carbon dioxide and this RUBP thing. The electrons that were carried, the energy from ATP and the electrons from NADPH will kind of power this. And basically, we're just popping off a carbon atom and incorporating it in with some hydrogens and oxygens to make a glucose. That's really the big picture of this whole process. So finally, turning carbon dioxide into sugar may sound fairly magical, but it becomes much more conceivable when you consider that CO2 and C6H12O6 or glucose have roughly the same sort of elements. The Calvin cycle just adds all of the extra elements required. Having said that, the just is still a major task requiring different enzymes all working in the correct order. And that is the big picture.